The second session speaker is Dr. Myra O. Villareal. Dr. Villareal obtained her BS Agriculture and MS Microbiology degrees from the University of the Philippines, Los Baños, and has previously worked for Peter Paul Philippine Corporation and the Ateneo de Manila University. At present, she is an associate professor at the Faculty of Life and Environmental Sciences of the University of Tsukuba, Japan, from where she obtained her PhD in 2011. She is interested in food functionality research, specifically in the use of natural products from foods for melanogenesis and anti-cancer functionalities. She has authored and co-authored more than 30 publications. Dr. Villareal?
Thank you very much, Professor Cadiz, for this opportunity to talk about one of our lab's research topics. Today, I will talk about the natural products effect on melanogenesis. Good morning, everyone. So we've all seen this somewhere, probably on our way to school or while we're in the mall. And we can all agree that the first thing that you'll notice is that the models, these beautiful models, have different skin colors. And although they have different skin color, the cells that's responsible for skin pigmentation, which is called the melanocytes, they're actually the same. In terms of number of melanocytes, every person has the same number of melanocytes. However, due to geographical location, such as for those people in Africa, they have darker skin color because they produce more melanin, while people in European countries have lighter skin color because of their location. So this is also not surprising. It's also not surprising when we hear of the high incidence of skin cancer melanoma among Australians, and that is because most Australians are originally from Europe and not from Australia. So let's talk about the cells that produce the pigment melanin. So that's the melanocyte. In this, the photo on photos A and B would just show you the location of these cells in the dermis or in the epidermis of the skin. So photo B actually have the MITF stained in order for us to identify which cells would be the melanocyte. And pigmentation, as we all know, is not unique to humans. We also observe pigmentation in Medaka goldfish, which is also used as a model in vivo as a model for in vivo studies on pigmentation. We also have pigmentation in dogs, in mice, as well as in polar bears. And mutations in the genes encoding key signaling molecules or transcription factors result in characteristic coat color phenotypes. So if you look at the mice here, it's probably not very important to most people, but if you're a mouse being chased by a cat, your color would spell your life or your death. So what is the melanin or pigmentation for? So primarily, as we know, we've learned this in school, is that the melanin protects us from the UV radiation. So we are aware that there are different wavelengths. We have the visible light, we have the UVA, UVB, and UVC. So if you notice, most of your protective lotion from the sun would be against UVB, and that is because the wavelength would actually be targeting the melanocyte at the specific layer of your skin. So it's very important that we get protection from UVB rather than from UVC or UVA, although that could also have some adverse effect. What other reasons do we have for studying melanogenesis? There are pigmentation disorders shown by this photo. This is melasma, or a hyperpigmentation disorder. Another one would be hypopigmentation, as shown in this right photo. And both pigmentation disorders have adverse effect on an individual, causing mental stress, for example. And aging is also affected. And as we age, we also observe hair graying. And that could also be another target for studying pigmentation. So at the center of the pigmentation studies would be this transcription factor called the microphthalmia-associated transcription factor, or MITF. So MITF is the master regulator of melanocyte development, function, and survival by modulating various differentiation and cell cycle progression genes. And at the same time, it's also an amplified oncogene in a fraction of human melanomas, and that it also has an oncogenic role in human clear cell sarcoma and other cancers. So MITF is between instructing melanocytes towards terminal differentiation or pigmentation development, and alternatively promoting malignant behavior. So how do we regulate MITF expression? So this photo would illustrate the regulation at the transcription level, A, and B would show you how we can regulate the MITF protein. So MITF gene expression is also under the transcriptional regulation of several transcription factors such as the beta-catenin, the PAX3, SOX10, CREB, and others. And the produced or the translated MITF or the protein could also be regulated by MAP kinases and CBP, P300, ubiquitination, and others. So this is a simplified schematic showing features of key regulatory pathways in the melanocyte lineage, which is also what I like to call as a map on how we can regulate pigmentation in mammals. So later I'll talk about natural products and how they can, the products, natural products that can regulate MITF. But here you can see how other natural products can also regulate MITF. So you have here the ligands or receptors that could be affected, the signaling molecules right after it, and the transcription factors, and the target genes. So centered to this would be the MITF, and MITF could basically inhibit pigmentation because it is the transcription factor for the melanogenesis enzymes, which are the enzymes that mediate the synthesis of melanin. So if you have a natural product and you want to check for the effect on pigmentation, you can basically check for the effects on these molecules. So just a brief information about how we produce melanin. So we need the tyrosine, which is also the precursor for other molecules such as neurotransmitters. But for melanin, we have the two types. We have the eumelanin and the phenylalanine. We need tyrosine, and the rate limiting enzyme for this is the tyrosinase, as well as the tyrosinase related protein 1 and DCT. Therefore, to directly affect pigmentation, you want to regulate this enzyme's expression, and upstream to that would of course be their transcription factor, which is the MITF. So in our lab, we use different plants, including medicinal plants and foods, and we study their effect on different biological processes, including melanogenesis. So natural products is the single most productive source of leads for the development of drugs. And do you know that about half of the drugs approved since 1994 are actually natural products based? And natural products is not limited to plants. They could also come from microbes and animals, as well as the synthetic or semi-synthetic natural products. So at present, we have this project that valorizes the bioresources in semi-arid and arid land for regional development. And one of the processes that we check is melanogenesis. And these are just some of the plants that we worked on. So one particular plant of interest is this Thymelaea hirsuta, and it is particularly significant to us because it's rich in Daphnan-type diterpenoids, which have been reported to have anti-cancer effect, and the extract itself is known to have an effect on metabolism. So two of the pure compounds that were identified in our laboratory by Myra et al. Would be hirsenes A and B, And the other plants that we also work on are in this slide, the Erica multiflora and C. spinosa. And the initial screening using these B16 melanoma cells. So for our research, we use pigment cells, one of which is this B16 murine melanoma cells. So just treating this mammalian cell with the extracts of these three plants, you can immediately see the effect on the cells melanin production. So in this test tubes photo, you can see that Erica multiflora EM made the lysate darker, and that means that more melanin were produced. And then the Thymilaea hirsuta had less melanin, and Capparis spinosa had also more melanin produced based on this. So that's an initial test that you can perform to see how plants extract could actually regulate melanogenesis. So we evaluated or we quantified the melanin produced by the cells after treating the cells with Thymilaea hirsuta. And you can see in this graph that there is a dose-dependent effect on the melanin production, but there was no effect on the cell viability. And if you look at the expression of the tyrosinase enzyme, you can see that there was also a significant decrease in the Thymilaea hirsuta tyrosinase expression. So, focusing on the pure compounds, hircine A and B, we also observed a dose-dependent effect. This is for hircine A, and this is for hircine B. And this is a positive control for melanin inhibition, arbutin, or something that is probably found in present in your whitening lotion. And also, what we also observed is that even after just 4 hours, the cell's morphology also changed. And that could also be an indication of differentiation in addition to the regulation of melanogenesis. So we looked at the expression of MITF as affected by the treatment of these pure compounds. And we can see that these two compounds from Thymilaea hirsuta also significantly inhibited the MITF expression as well as the enzymes expression. So this was also accompanied by a decrease in the expression of the protein of tyrosinase, the enzyme. And we also evaluated the mechanism of the effect of just for hircine A using DNA microarray, and we can see that most of the genes that were significantly regulated were either MITF targets or genes that actually regulate also MITF. So these are in this photo, and you can also learn about this more on this article that we have published on this topic. And just going back to the differentiation effect, you can see that hircine A also changed the way the cells would grow in the culture plates, as shown here. So other products that we also work on that also has an effect on melanogenesis or MITF would be this 3,4,5-triorthocophylcanic acid or TCQA, which you can find in coffee, and purple sweet potato. You can see in this photo of this mice that the coat color was actually darker than the control, and also the growth was also faster. And we can also see that MITF was significantly increased by TCQA in both at the transcriptional and at the translational level. So another plant that we worked on is this Tara, and its tannin or Tara tannin also inhibited or rather promoted MITF. So Thymilaea hirsuta inhibited MITF, but triglycylcanic acid and Tara tannin promoted MITF and promoted pigmentation, as you can see here. So this is a summary of how Tara tannin modulated or rather regulated MITF through these other upstream molecules being targeted by Tara tannin. And just to summarize, natural products may be used to regulate MITF for both pigmentation-related or cancer-related studies. And there is also a need to focus more on the earlier signals to determine how natural compounds regulate MITF expression. Okay, thank you, and I'm ready to answer questions later. I would also like to thank our laboratory head, Professor Estrada, and the 2 students who worked on tyrosine and TCQA. That's all. Thank you.
Thank you, Dr. Villareal.